Hi, everybody. Let's wait for people to roll in, finish their breakfast, lunch, I'm guessing. And so, good opportunity as ever. If anyone has any questions about the mini lab report in terms of content, grading, so on and so forth, now's a good time to ask.
So you said to try to have a rough draft to you by Friday or something to give you some time to look it over and give us some
Ideally,
feedback. Okay.
yeah. And then I can fit it into free time that I have over the weekend and get it back to you for sure by Monday because it's due on Tuesday midnight. Now, if you do get it to me on Monday, then I'll endeavor to get it back to you before the end of the day so you can work on it. But obviously, depending on when you get it to me, there's no guarantee I can do that. So but it's also, it's funny. No one likes deadlines, right? Because it means you have to do stuff. But the alternative is having to make sure you do this stuff yourself. And so in some ways, it's better to view deadlines as like an aid to, keeping on track and getting everything done rather than a drag. Not always easy. It's obviously easier for me to say that because, I'm not the one that has a deadline. Okay, anybody else have any other questions? Hopefully the material online on Blackboard is what you need. But if not, then obviously let me know. Cool. Okay, so that lab report is really the kind of the end of that particular series of experiments. But really, in a way, you can consider that work as being kind of like the preliminary work that you would do to characterize a gene. And so, I mean, obviously, in this modern era of having genome sequences available for lots of different organisms, it's a little different. And particularly now that CRISPR-Cas technology is becoming more widespread, right? It's not just in certain key organisms, it's in more of the other. Yes, that's right, Adam. Rough draft by tomorrow at some point. Final draft Tuesday at midnight by email. Yeah, so anyway, if you, for example, this wasn't quite what I did, but what my last postdoc supervisor did for her postdoc, well, she did a large genetic screen and she was looking for mutants that displayed a particular phenotype, which is called a connection of gonad phenotype. So essentially she was looking at genes that caused issues with egg laying in nematodes, right? Because she was interested in the developmental biology of that process. And so, she got a whole bunch of different mutants. I can't remember what kind of mutagenesis she used, probably ethidium bromide or something like that. Oh, not ethidium bromide. Doesn't matter. But anyway, you basically got a bunch of worms that look different, right? Or in the case of what we're going to be talking about, flies that look different. And so one of the central tenets of genetics is that those visible differences are caused by underlying molecular mechanism, right? Changes in the DNA which are heritable, which change the phenotype you see. And Adam, no, I think that's somewhat unlikely. Obviously, we're not going to look the same today as we do in 100 years. Actually, South Park had a funny episode once where there was time travelers came back from the future, and they were all kind of like a light brown color, and none of them had any jobs because they've all been made unemployed by robots. So that was that was one. They all look kind of homogeneous, right? Because of mixing between different groups of people around the world. So I'm not sure. Maybe that's one possibility. I mean, the funny thing is that again, X-Men and the like, kind of those stories are all predicated about the idea that mutations can generate like really kind of novel, awesome phenotypes, and that's typically not the case. So this is kind of more of a genetics question. We'll get back to the lab stuff in a second. Typically, mutations in single genes which are important for something result in lack of that function, and if that function is important enough, then lack of you, right, or some kind of congenital disease. The beneficial mutations, which are few and far between, those need to be selected for. All right, those alleles that are in the population need to be selected for over a really long period of time before you ever see even like a small difference, right? So we're thinking about, you know, as humans, we tend to think about on the generational scale, right? You can think about your grandparents, you can think about your children, right? It's kind of like 4 generations is about longest time span we can think of, 100 years, actually, kind of like what you put. But just as a case in point, there's a mutation which they believe gave resistance to smallpox, and in doing so, pardon me, confers resistance against HIV, right? It's a mutation that's somewhat common in Northern Europe. And that allele is present at about 10% frequency in the whole population. And they think that that took about 800 to 1,000 years of smallpox, which by some estimates killed a quarter of the population of Europe over that time, to get to 10% frequency in the population, right? So what we see as changes between us and other species that we share a common ancestor with, like chimpanzees, for example, those changes are a result of accumulations over millions of years. And so 100 years is, even if there were going to be X-Men, which, even though I like X-Men as a series, I think it's super cool. Pragmatically, I don't think we'll get there. I think like the rise of artificial intelligence, and I'm more of a Terminator kind of, this is what will happen in the future kind of thing, you know, taken over by robots, you know, Matrix type stuff. That's kind of my view of the future, rather than humans becoming superheroes and stuff. I guess the more pessimistic side of things. Anyway, that's kind of a little bit of a tangent. But so essentially what you need to do is you need to link that phenotype, like what you can see, with the underlying genetics, right? And so that's a central tenet of genetics, is that a change in the DNA sequence typically is what is required for you to see a change in a phenotype, right? Something visible. But until very recently, we're unable to actually go from genotype to phenotype, right? In terms of creating changes, like targeted mutation, or what they call reverse genetics, is a very, very recent advance in genetics. The classic way is just to screw these organisms up in terms of their DNA sequence and then see if you find something interesting, right? That was what happened with the flies with Thomas Hunt Morgan. I think he actually found a naturally occurring mutation. I don't think he induced it. I think he just looked at a lot of flies and found one with white eyes. And was like, that one looks special. Let's keep this one. And worked with it from that point. So once you have that phenotype, sorry, I don't have enough time to swallow my lunch. Once you have that phenotype, how do you figure out what its genotype is? Now, what the genetic cause is? Well, the first thing you do is figure out, well, is this phenotype caused by just one gene, right? Do my mutant alleles that generate this phenotype, are they all caused by the same gene? That kind of gets back to the idea of complementation that we talked about a little while back. But even if we figure out it's down to one gene, well, where is that gene? What chromosome is it on? What bit of DNA can we narrow it down to? And so essentially that's what you would do with that linkage experiment, right? So if you were working in an organism that didn't have a genome sequenced, you'd be like, okay, well, we've got 6 chromosomes. Which chromosome is it on? Okay, it's on chromosome 2. Well, where on chromosome 2? And then you kind of narrow it down further and further until you get into like the rough neighborhood, essentially, of that mutation. Again, doing those exact same kind of linkage experiments that we did for figuring out the distance between yellow and white, right? That's exactly the same kind of work. Now, that's kind of old school genetics. Now we go into new school genetics, where we're trying to figure out, well, what's the sequence change that resulted in that phenotype? And so that's essentially what we're going to be doing for the next, well, actually till the end of the semester for the lab. Yeah, there we go. And so really, actually, I can just share that with you. So really the purpose of this module, and this actually got accepted by American Biology Teacher Journal, ABT, which is a pretty good teaching journal. Dr. Page and I wrote this up as a kind of a package type paper. Here, if you want to do this kind of work, here you go. Here's all the stuff you need. So this will actually come out and print in 3 to 4 months, I guess. Been a little while now. And so really what the purpose of this is kind of for you to imagine, okay, we've got this phenotype, we've got a definite mutant, right? This phenotype is inherited, right? It's not just random. If we do classical genetics, we can figure out what the mode of inheritance is. Is it sex-linked? Is it autosomal? Recessive, presumably, but might not be, right? How do we go from that to figuring out what's the mechanism, right? Because that's what's interesting, well, at least to me, right? What changes, what happens to cause this particular phenotype? And so what we're going to go through over the next 6 labs is really the exact same things that you would go through if you were doing that kind of experiment. It's the same thing that I've done in the past when I've isolated a particular gene involved in a process, like one I did for my PhD, gene called DAF7 in a parasite, right? You're going to be doing exactly the same kind of stuff, the same theory, right? And so generally what you do is, well, you have to get DNA out of your organism first, right? That's going to be what you're going to use. You need to make sure that's good DNA. Then when you have that, you're going to try and amplify that DNA to essentially go from isolating the DNA from your organism to isolating the bit of DNA that spans where you think your mutation is, right? Or if you're just trying to find a gene from scratch, like where you think your gene is, just so you can figure out what the sequence is. Kind of makes me feel a little bit old talking about kind of stuff I did for my PhD, because that was a long, long time ago. And it was kind of back in the old school days before genomic sequences were available, which is kind of crazy. So once you have that, you need to clean that fragment up, right? Now, the aim of PCR or polymerase chain reaction is really to amplify a small bit of DNA so you have enough of it to work with. And then you send it off for sequencing. Someone else does that for you. You give them like $8 a read. And then you have to analyze it, right? So typically the first thing you do is you kind of clean up the sequence. You get a reference sequence, right? So in this case, it's going to be the fly genomic sequence. In my case, it was a series of related organisms, right, that had that gene that I was working on that I wanted to align mine with and see if it was a close match or not, right? See if it was similar. And that's how you figure out this phenotype is caused by this gene. And quite often, the nature of the mutation will give you information as to why you observe that phenotype. The actual sequence you get will potentially, if you use the right bioinformatics software, give you an idea of what actually that protein is, right? Because you can search databases to look for similar proteins that may have been studied by other people. So really, even though this is going to feel a bit weird because we're doing this all kind of online and not in person, really what you're doing is you're going through the same steps that people around the world are going through right now to isolate and characterize genes that are responsible for traits that they're interested in. Obviously, you're not doing this from scratch, right? We're working with something that we already know just because the tools are a little easier, right? You're not so much in discovery, you're more in learning the concepts that are involved. And so what we're going to be working with, oh, and also this last part is using a bioinformatics software program called UGene. It's not a trivial piece of software to use, but it is very much the same as you would use in a lab unless they pay for a different copy yourself. And it's actually pretty cool. I was talking about this particular lab with the dean of the College of Arts and Sciences, and she was impressed that you get such a cool exposure to current technology in genetics lab. She said that they didn't do anything as advanced as that at Texas State. So that was kind of neat to hear that. Obviously, the downside is bloody difficult to use, and so there will be a few hiccups, but you'll get better education than people at Texas State, at least for this lab, which is always nice. So the thing that we care about is one of the phenotypes that we've already looked at before, and that is the yellow phenotype. So if you look at normal flies, right, that might be flying around your fruit bowl, for example, they are around mine, you'll see they're kind of dark brownish. I mean, they actually like look a little bit black, but if you look at them under a light microscope, they're kind of brownish with these dark bands on their butt or their is that their thorax? No, that's their abdomen. There you go. I'm not very good at fly anatomy, even though I've been working with it for ages. And those bands look different as to whether they're male or female, right? So the males are smaller, the bands at the tip kind of run together, so they're darker. But all of this is much paler, right? So you can kind of see through the wings. Those bands are just about present. You can see them more kind of down here, but they're much paler, right? And so these are called yellow flies, right? And the wild type is either wild type body or brown body flies, right? And so through the genetics that we've done, we know that yellow is on the X chromosome, right? It's closely linked to the white gene, so we kind of know about where it is. What we're going to be going through is the process by which we figure out what the gene is and what is causing this yellow phenotype, right? And so yellow is actually, it's such an interesting, it's an interesting gene because it's what's called pleiotropic. I'll write that down for you because it's kind of hard to spell out. Oops, come on. And what that means is one gene affects multiple processes. And so in the case of the yellow gene, the most obvious one is pigmentation. I mean, yellow phenotype is because they're yellow, right? But another one which is really curious is that it also affects male mating behavior. If I can spell that. And one of the reasons is from the reading that I've done is that these sex combs, right? So these are on the forelegs of the flies, kind of about here. If this was a fly leg, so it's not. Kind of about here, and they actually look like combs, and they're used to grasp onto the lady flies during copulation. And so in yellow mutants, these combs aren't fully developed. Right? So yellow males aren't very good at reproducing with females. And there are a few other kind of related courtship behaviors, a bit weird, don't know why. And so yellow actually encodes a dopamine converting enzyme. Come on. Which is kind of weird because we think of dopamine as being a neurotransmitter, which it is, but it's also the basis of a lot of pigments. It's part of the pathway of generating pigments like melanin, which is kind of weird. But not really much more is known about it than that, which I always find kind of strange, given that this is such a well-studied, gene and phenotype. Anyway, so we're going to be working with this, and what you're going to be doing is isolating, well, obviously not in person, but we'll be going through the concepts and the theory of isolating the DNA, amplifying part of the yellow gene, and then for the U gene lab, which is the last two labs on the schedule, you're going to be comparing the sequences that you get, which we have a big folder of, with the wild type and mutant yellow reference genes, right? To basically mutant reference Drosophila genes, yellow genes, that's right, to see how they compare, see where that mutation is. And that's pretty much it. So most of these, not today's lab, but following labs have assignments to go with them, the same as the first few labs did for the course. They're really based around kind of interpreting data and doing calculations, bit of the concepts and the knowledge about what you're doing, similar to what you've been doing before, but specific for those particular procedures. The only thing that you're missing out is actually the mechanics of pipetting stuff out and centrifuging things and stuff like that. And hopefully you'll get that experience in other courses as you go through your biology degree here. And that's pretty much it. I don't really have much else to talk about today. I really wanted to give you the overall view. But one reason why I really like teaching this, even teaching it online when we don't get to do the stuff, is because this is exactly what you would do if you were working on an unknown gene in a research lab, right? So these are exactly the kinds of skills that you need. So hopefully by kind of going through the different steps in this process, when you do encounter this kind of work, if in other courses, or hopefully when you go on to careers in research or equivalent, you'll be like, oh, right, yeah, I remember that. You've got to do this, this, and then this. This is easy. We covered this in genetics, right? And even if you don't go on into research, it gives you a good taste of the kinds of work one can do if you are studying this kind of genetics. Cool. That's pretty much it from me. It's a real short one today, which probably no one is going to complain about. And you can if you want, but it's not going to make any difference. It's still short. So yeah, that's pretty much it. Again, get me your rough drafts, ideally by tomorrow, and at the very, very latest by Monday, and not late on Monday either. And I'll get them back to you with comments, basically telling you where you need to improve to meet the rubric. So the closer you hew to the rubric, right, literally ticking off those different things, the less work you'll have to do when you get your comments back. That's pretty much it. Anybody got any questions? Nope. Cool beans. Well, enjoy the rest of your afternoon. It's always nice to have an early break. And weekend too. Happy 4th of July. Although it kind of pains me a bit to say that as a Brit, but you know, that's old history, right? No one's going to hold it against me. I hope. Cool beans. All right. Have a great weekend, everybody. And avoid Chernobyl. Yeah, no kidding.